All right, so we covered translation and how translation takes an mRNA sequence and converts it into a polypeptide chain. And we covered kind of how proteins can look different based on their environment or based on the amino acids in the chain. But we haven't really covered yet how we get from here to here. So I want to take a minute in this video just to talk about the 4 levels of protein structure. We have primary, secondary, tertiary, and quaternary structure. And you could also represent those as primary, secondary, tertiary, and quaternary. Primary structure is going to be just the sequence of amino acids in the chain. Primary structure is what you end up with at the end of translation. And let's watch a little video showing that in more detail, and then we'll come back to this slide.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay, and then that primary chain, remember this thing can be hundreds to thousands of amino acids long. And so it's not just going to hang out in this one sort of long stretched out chain of amino acids. It's going to start folding back on itself. And that's when we end up with secondary structure. And so this chain of amino acids is either going to kind of form this helical shape or fold into this accordion shape that you see here. We call these an alpha helix and a beta pleated sheet. And secondary structure is what gives proteins their strength and flexibility. You can think about an alpha helix as kind of acting like a spring. Like if you were to pull a spring, you could stretch it, but it would snap back really fast. So alpha helices give proteins their strength. And then if you think about the beta pleated sheet, that's kind of like an accordion where you can pull the pleats apart pretty easily and push them back together pretty easily. And so beta pleated sheets give proteins their flexibility. And let's look a little bit in more detail at that folding process.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself or where 2 regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
So as you just saw in that video, this secondary structure is determined by hydrogen bonding in the backbone. Remember that backbone has amino groups and carboxyl groups made of polar covalent bonds. So you end up with partially positive and partially negative atoms that are attracted to each other, resulting in hydrogen bonding between the backbones of amino acids in the same polypeptide chain. Tertiary structure is where we start to think about the R groups rather than the backbone. So primary and secondary structure is all backbone, either linking the backbone together via peptide bonds in primary structure or hydrogen bonding between the backbone in secondary structure. And then tertiary and quaternary, these are going to be between R groups. And tertiary structure, this is achieved through folding and interactions between R groups with each other. And so we will see negatives attracted to positives, or negatives repelling other negatives, or van der Waals between R groups that are both neutrally charged, or there's one new type of intermolecular interaction that we did not talk about in Unit 1 that we do see in protein folding. So let's take a look at that here.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, so as you saw there, stuff that should be familiar to you from Unit 1 is hydrogen bonding between partially charged atoms in neighboring R groups. So a partially positive hydrogen attracted to a partially negative oxygen or other negatively charged atom. Ionic bonding, where a full negative is attracted to a full positive. Or van der Waals, where neutral charges are attracted to each other. So those are Unit 1 concepts. The only new one here is the formation of disulfide bridges. In proteins, some R groups contain sulfur, indicated by an S, and if those R groups end up near each other, those two sulfurs can come together and bond to each other. And this type of tertiary structure is really common in hair, and it's what helps to determine hair texture. So somebody that has really, really curly hair has a whole lot of disulfide bridges, and somebody that has very straight hair doesn't have that many disulfide bridges. All right, let's play with this a little bit more here. Let's see. All right, I'm going to post the link for this below this video so that you can play with it as well. But this is basically a little widget that lets you take a polypeptide, take a primary structure of a protein. All of these here are the abbreviations for the different amino acids. And then you can choose to drop it in water or drop it in oil. You can choose if you want this protein chain to be made up of mostly hydrophilic or mostly hydrophobic amino acids. And you can watch what happens. You can watch it go all the way through to a tertiary folding. So let's go ahead and see what happens with this amino acid if we drop it in water, indicated right here in the drop down. Okay, so I very purposefully didn't tell you what green and yellow referred to yet. Based on how this protein is folded, do you think the yellow amino acids are hydrophilic or hydrophobic? Remember, we dropped it in water. Yeah, so all of the yellow ones here are going to be hydrophobic. They're kind of hiding together in the middle of the protein. And the green ones are hydrophilic. They're like happily hanging out towards the outside, forming hydrogen bonds with the water. If we go ahead and, let's reset. If we go ahead and make the entire chain hydrophobic, what do you think is going to happen? We're still in a watery environment. Yeah, it's all going to clump up. This would be just like dropping a drop of oil into a cup of water. So they're going to form van der Waals interactions with each other, and they're going to avoid the water. What if we make the whole thing hydrophilic, or mostly hydrophilic? Yep. Interesting, right? So since the hydrophilic amino acids are happily forming hydrogen bonds with water, this thing doesn't feel the need to clump up as much as it did before because the hydrophobic amino acids don't all need to get together in the middle. They're more dispersed. They're not really all together here. If we go ahead and flip it, making it mostly hydrophobic, now it's going to come back together. If we go back to a random mix, again here you can watch how the uncharged amino acids always find their way to each other, or mostly find their way to each other, and the charged amino acids happily hang out towards the outside. What if we take, I'll reset it, what if we take a totally hydrophobic chain and we put it into oil instead? What do you think is going to happen with this one? Yeah, it totally spreads out. So now, because we're putting non-polar amino acids into a non-polar environment, they are going to form van der Waals with each other and form van der Waals with the surrounding oil. So they're going to be quite happy. What if we switch this chain to totally hydrophilic? So now all of these partially and fully charged amino acids are more attracted to each other than they are to the surrounding oil, so they clump up. They clump up just like a hydrophobic chain did in water. And so again, I encourage you to play around with this in the link that I'm going to put below this video. But for now, let's go back and talk about quaternary structure. All right, so the last level of structure is when multiple subunits of proteins come together to form the final protein. So take hemoglobin, for example, that we already talked about. Hemoglobin has 4 subunits, and so that was 4 pieces of primary structure protein. That was 4 individual polypeptide chains that each went through secondary and tertiary structure and then came together to form the quaternary final protein. That was 4 different proteins coming together to form a final functional protein in the cell. So quaternary structure is multiple polypeptide chains. Quaternary structure is multiple chains of amino acids.
Some proteins consist of 2 or more polypeptide chains. The 4th level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
Okay, and so in the next video, we're just going to talk a little bit more about different types of quaternary structure and how you can affect protein structure in different ways. And so we're going to end up with 2 basic types of proteins. We're going to end up with globular proteins like hemoglobin and then fibrous proteins like collagen or like keratin in your hair.